All right, Chapter 13 goes over genomics and biotechnology. So when we talk about genomics, it's the field that compares the entire DNA content of all different organisms. So we know the genome is the full genetic information of an organism. When we talk about genomics, we're comparing the genomes of all these different organisms. When we talk about DNA sequencing, that's the process that allows scientists to read the sequence of each nucleotide in a DNA strand. In DNA sequencing, a fragment of DNA is going to be amplified so that there are thousands of copies. After that, the fragments are going to be mixed with copies of DNA polymerase, primers, and a supply of nucleotide bases. This is what allows us to add new nucleotides to make new copies of that DNA. Also, in this mix, we're going to add 4 different chain terminating tags, and these are going to be the spot that that DNA copy stops. So once that chemical tag is inserted, that's when we're going to be at the end of our stretch of DNA that we're copying. In order to do this, we apply heat, and that denatures the double-stranded DNA. That's what pulls our double strand apart into 2 open strands with exposed nucleotides, so that we can bind new nucleotides and create new copies. In that process, DNA polymerase is the enzyme that is synthesizing our complementary strand until the chemical tag is added. Also, because of the relatively low concentration of our chemical tags, compared with the high number of nucleotides we've added, the mixture will contain a series of double-stranded DNA fragments of varying lengths, because it's all going to depend on when that chemical tag is inserted. The fragments of our DNA are going to be separated according to size, and we do that by using gel electrophoresis processes. The fragments will become arranged in an order, and the sequence of nucleotides can then be read 1 nucleotide at a time. Our DNA sequencing is now automated, and it can actually handle a really large number of samples. This is what has made it possible for us to sequence really large eukaryotic genomes, like the human genome. So when we look at this process, the first step is our primer extension reactions, where we add the primer and the nucleotides that will be put in place for our DNA copies. So in step 1, DNA is sequenced by adding complementary bases to that single-stranded fragment. DNA synthesis will stop when the chemical tag has been inserted instead of a nucleotide, which is going to result in different sized fragments. So it's like the chemical tag will pair with a nucleotide, but is not a nucleotide itself. So that's going to halt the copying process or the placement of new nucleotides. In the second step, the DNA fragments of different lengths are going to be separated by gel electrophoresis, and the smaller fragments are going to travel further down the gel. Our third step, we will use computer scans to analyze the data from our gel electrophoresis. The smallest and largest fragments are going to be displayed in a series of corresponding colored peaks based on their size. And that's going to indicate which nucleotides are present. After that, the data from an automated DNA sequencing run will show the nucleotide sequence for a small section. And in this example, we have an example of a plant's genome and its sequencing. So you can see here the size of the colored peak after we have analyzed our gel electrophoresis is indicating which nucleotide is placed there. Here you can see these are the peaks indicating thymine, these peaks indicate adenine, these black peaks indicate guanine, and then here you can see cytosine. So that's telling us exactly which nucleotides are appearing in our DNA strand and in which specific order. The publication of the sequence of our entire human genome actually just came out in the year 2000 on June 26th. And the human genome contains a shocking more than 3 billion base pairs in the entire sequence. It's estimated that there are only 20,000 to 25,000 protein encoding genes. So of all those 3 billion base pairs, there are only about 20,000 to 25,000 genes that will code for proteins. And that comes out to just 1%, actually less than 1% of the human genome responsible for coding our proteins that we need. When we talk about genomes, here are some interesting comparisons. So, as far as humans, we have the first large genome to ever have been sequenced, and the number of transcribable genes is actually far less than they anticipated, because much of our genome is actually occupied by those repeated DNA sequences, or non-coding portions of our DNA. When we are comparing the human genome to the chimpanzee genome, there are very few base substitutions between the chimp and human genomes. It's actually less than 2% difference. But there are also many small sequences of DNA that have been lost as the two species have diverged, which has some significant impacts on the phenotypic expressions of our genes. When we look at the mouse genome, about 80% of mouse genes have a really functional equivalent in our human genome, and large portions of the non-coding DNA of mice and humans both have been conserved. Overall, rodent genomes like the mouse and the rat appear to be evolving more than twice as fast as primate genomes, like the human and the chimpanzee. Another interesting point here is that the pufferfish genome is only 1/9 the size of the human genome, yet it contains 10,000 more coding genes. So even though it is such a smaller length in size of nucleotides, it still contains 10,000 more coding genes than we have. An interesting point when we're comparing these invertebrates is the fruit fly and the mosquito. When we look at those side by side, the similarity between these two species is about equal to the similarity between humans and pufferfish. So it's not always really intuitive how these genomes are going to show up. Because, you would assume there's a lot less difference between a fruit fly and a mosquito than there is between a fully functioning human and a pufferfish. But that's actually not the case. We have about the same differences in our genome as the fruit fly does to the mosquito. Here are some plant genomes that have been sequenced. The rice genome contains only about 13% as much DNA as the human genome, but it also has twice as many genes as we do. Like the human genome, it is also very rich in repetitive DNA. When we're looking at the cottonwood tree, this is the fast-growing tree that is widely used by timber and paper industries, and its genome is 50 times smaller than the pine tree genome, which is 1/3 heterochromatin. The number of genes is going to be much less than was originally expected based on unique mRNAs found in human cells, because our first guess of how many genes would be in the entire genome is based on all the different combinations of messenger RNAs that we know code for proteins. But in eukaryotic DNA, those protein encoding segments are called exons, and they are interspersed among what we've previously mentioned, the non-coding portions of DNA called introns. We know that alternative splicing of those exons accounts for the fact that about 25,000 genes can actually encode 4 times as many proteins. When we talk about alternative splicing, that's what we mentioned in chapter 12, where even though we have transcribed one section of DNA and created a primary RNA transcript, we're going to cut out those exons so they're separate from the non-coding introns. The alternative splicing comes in at that point because we could rearrange the order in which we're putting those segments of exons so that now we're coding for entirely different proteins. Some of our chromosomes are going to be packed with genes, and others could contain relatively few genes. It just depends on the stretch of DNA we're working with. When we compare genome size, you can see humans over here with a smaller genome size than chimpanzees, monkeys, dogs, cows, rats, mice, chickens, and pufferfish, but a significant higher amount of nucleotide base pairs in pufferfish, rice, and also the cottonwood tree. So just because they have more nucleotides does not mean they're more complex organisms. A lot of times simpler organisms have longer stretches of DNA with a lot of repeated sequences or a lot of introns, a lot of non-coding portions of DNA. When we talk about those portions, there's really these four basic types that make up all of the DNA that is not coding for proteins. And so these account for 99% of the DNA in humans. Only that 1% is going to code for proteins. The first of which is our non-coding DNA within the genes. That's the portion we call introns. We also have structural DNA, which is present just for the structure, for arranging our tightly coiled chromatin in the right distance from the genes. It also reduces the chance of mutations based on genes being so close together or separate genes that should not be interspersed whenever the exons are rearranged in alternative splicing. If we have a greater distance between them, either by these structural DNA or repeated sequences, then that benefits us. The next kind of non-coding DNA are the repeated or duplicated sequences. We tend to call these simple sequence repeats, and they are duplicated sequences that are scattered about on chromosomes, again with the benefit of providing some distance between really important genes that should not be spliced together. We also have transposable elements, and these are bits of DNA that jump from one location on a chromosome to another location on a chromosome. So they are able to move. So when you're trying to think which one that is, you could think trans like transit, when you're in transit, when you're on a train, when you're traveling somewhere, like these transposable elements. They can be moved around on the DNA strand. So here you can see we've got only 3% of our DNA made up of repeated sequences. 1% is coding for all of the proteins that we need to make. 7% is made up of those duplicated sequences that are not SSRs. Then we have 20% as structural DNA. 24% are introns, which are the non-coding portions actually inside the gene that will be spliced out. And then 45% of all of our DNA content is those transposable elements. When we talk about genetic engineering, that's whenever we are moving genes from one organism to another. So the first stage in a genetic engineering experiment is going to be to chop up that source DNA. We have to be able to take out the specific gene that we want to move. So we have to cut it out of the strand. And then we have to copy it, because we have to have a copy so that we can transfer it. We do this using restriction enzymes. They're also sometimes called restriction endonucleases. And they will bind to specific short sequences on the DNA and then make a very specific cut. That sequence is always going to be symmetrical, and the cut will generate DNA fragments that are kind of sticky, because the incision made by that restriction enzyme is going to be to the side of our DNA strand, so that sugar phosphate backbone. We're not cutting in the middle where those hydrogen bonds are, we're on the side where our sugar phosphate backbone is. So that's going to create strands that have an affinity to bind to other segments of DNA. DNA from another source, whatever source we want to combine this cut DNA with, will be cut with those same restriction enzymes, and then it's going to end up with the same sticky ends that are prepared to bind. Those ends can be joined together by just adding the enzyme ligase, which usually exists in our cells for the purpose of sealing newly placed nucleotides in place. But now we can add it and help to synthesize our recombinant DNA. By adding ligase, we can seal those two separate cuts of our DNA segments together and have a whole new strand of DNA. So these restriction enzymes are really the basic tools of genetic engineering. And here's a look at how that process works. So we have our restriction sites shown here, which are going to be where the restriction enzymes show up. The restriction enzyme will cleave the DNA at those sites. We're going to end up with these sticky ends that we mentioned. We will then add DNA from another source that's cut with those same restriction enzymes, and then ends up with the same sticky ends. There will be a natural affinity between these strands because of that, and because of the way that their nucleotides align with each other as well. Then we add DNA ligase, and suddenly we have recombinant DNA, where we have inserted new genetic information into a strand that has old genetic information as well. So in this process, we have a need for complementary DNA, and that's usually named by this lowercase c in front of DNA. Recall that eukaryotic genes contain both those exons and introns, parts that code exons and parts that don't, introns. So before the gene can be translated and expressed, the introns are going to be cut out. We know that. It happens in the nucleus. We get that primary RNA transcript. Then alternative splicing happens where we cut out the introns and we splice together the exons in whatever order is needed for the specific protein we're trying to make. The enzymes stitch together the remaining exon fragments to form our mRNA, messenger RNA, which is then eventually translated in the cytoplasm. But also, bacteria lack the enzymes to remove introns, and we know that it's desirable for genetic engineers to transfer an intron-free copy of the gene into bacteria. Whenever we're forming that complementary DNA, we have to be able to obtain DNA without introns. Genetic engineers will do this by isolating the processed messenger RNA corresponding to a particular gene. So it makes sense. Instead of us trying to make a copy of this long stretch of DNA the way that it shows up, the only way to really get around that and not have to go cut all of the introns out ourselves is to start with the messenger RNA that already has those sections of introns cut out, and then go backwards and create a DNA copy from our messenger RNA template. We do this using the enzyme reverse transcriptase, which is pretty appropriately named because we're doing transcription in reverse. Instead of taking a DNA strand and making an RNA copy, we are using our messenger RNA as a template to create our DNA strand that doesn't have any of those introns in the way. And that's what we call complementary DNA. So that's just the DNA we've created based on an mRNA template so that we don't have introns. You can see that happening here. We've got our regular stretch of eukaryotic DNA. This is a gene that includes all of the non-coding portions and the coding portions. After transcription, we will have that primary RNA transcript. The cell will naturally undergo the process of alternative splicing where we cut out our introns and splice together the exons in the correct order. Once we have that messenger RNA, we can add reverse transcriptase and then end up with a perfectly matching complementary DNA strand that is going to have all of the information of those coding genes and none of the information from the introns. After that, we have successfully copied this strand using reverse transcriptase, and that's the general process of creating our complementary DNA for whenever we need to have a very specific copy. In in biotechnology and genomics, we also have the process of DNA fingerprinting, which is a very revolutionary technique, and it's used often in forensic evidence. I'm sure you guys have seen that in TV and in the news. This process uses probes on DNA samples that have been cut with those same kinds of restriction enzymes or restriction endonucleases. The probes are going to be complementary to those unique DNA sequences found in the non-coding regions of human DNA that are really highly variable among individuals. So a lot of the usefulness of our non-coding DNA related to DNA fingerprinting is that's where the most of our variety is. And that makes sense because if we're all trying to code for the same proteins, then our exons are going to be present in each one of us. But the random assortment of nucleotides in our non-coding regions of DNA are what provide a sort of fingerprint that is unique to that individual. Each band will represent the position of, whenever we're looking at this DNA fingerprint, it's going to be read on an audiograph with rows of parallel bands on an X-ray film. Each band on that film is going to represent the position of a DNA fragment that complementarily binds to one of the probes. And that's what's really giving us the sequence. So our DNA fingerprints have been used in courts of law since 1987. And while an individual DNA fingerprint is not 100% accurate, it's definitely accurate enough that we can rely on it just as much as fingerprinting, just from our fingers. And it is significant enough that we are able to use it in a court of law as proof. This can be used to convict or to clear a suspect. And this is from any source of DNA, whether it's from the hair or a speck of blood or semen. All of those have your cells, which have a nucleus, which carry your genetic information. So it doesn't really matter which cell we're taking the information from. They all have that same genetic information in the nucleus. So even though this process is definitely significantly enough, it's efficient enough to prove the difference in an organism based on the genetic information we find in the sequence of nucleotides in the non-coding regions. But that doesn't mean that no problems could happen. So if you're carrying out the laboratory analysis of this DNA and you do something wrong in that process, you could still end up with inaccurate results based on the way that that information was processed, even though DNA fingerprinting itself could be accurate enough to discern between two samples of blood or hair or semen. If there is a problem in the processing of that DNA, it could be enough to throw off the accuracy of those results. One of the cases that chapter 13 discussed in our book was the Warney case. And this is about Douglas Warney. In 2006, he had been in prison for 10 years for the murder of a man named William Beeson. The main evidence that proved him guilty to get him convicted had been the claim that he had said information about the murder that no one could have known unless they were there. But Douglas applied to the Innocence Project in hopes that they would provide a DNA analysis after he'd been in prison for 10 years. The judge actually overturned his plea to re-examine the DNA and just basically said, no, we're not going to look at it again. But somebody went ahead and had the test run against the judge's order. The DNA ended up matching a man who was imprisoned for the murder of his landlord just weeks before William Beeson was killed. Whenever they went and questioned that man who was already held on life without parole, they questioned him and he readily admitted to the murder. He said, yeah, that was me. And as a result, Douglas Warney was then set free. Whenever you hear the term PCR, that's referring to polymerase chain reaction, and this is a method that we use to produce millions of copies of DNA from just one single strand. When we do this, we have a double-stranded DNA fragment, and in order to separate those two strands, we denature it with heat. So we heat it up, that breaks the hydrogen bonds that's attaching all of our nucleotides, and now we've got template strands. Each of these strands is going to be copied by DNA polymerase, which is an enzyme that we will add so it can fill in the corresponding nucleotides to our exposed DNA templates. And then this cycle is just repeated over and over again. Every time we repeat the cycle, we're going to double the number of DNA copies we've made. You can see that happening here. Our first step is denaturation. That's where we're going to heat up our DNA strands so that they separate. Here you can see they're together. We heat them, they separate. Then we have to add primers, because if we are going to add DNA polymerase, it has to have a primer to know where to start. So we add our primers. That's going to tell DNA polymerase where to start adding nucleotides. After that, we add DNA polymerase and all of our free nucleotides, adenine, thymine, guanine, cytosine, so that they can be placed. DNA polymerase will start putting nucleotides in place on our new daughter strand of DNA. After that, we can heat the strands so they separate and cool them again so that they rejoin after a copy has been made, and that will be a single cycle. We continue to repeat that heating and cooling process so that these strands can be joined together, and we can continue to double the amount of DNA strands that we have with each cycle. A lot of the promise of genetic engineering actually lies in treating illness. So that's one of the main goals around genetic engineering. And there are many genetic defects that occur because our cells fail to make critical proteins that we really need. One example of that is diabetes. Diabetic people cannot control their blood sugar levels because they don't have a protein. They can't make it. They don't have the gene to make insulin. But through genetic engineering, through genetic engineering, the genes encoding insulin have been introduced in bacteria, which can then cheaply produce large quantities of protein. So basically, we insert that gene into bacteria that then start to produce insulin, and then we harvest that insulin and provide it to humans that are not able to produce that protein at all. Another example of this is factor VIII, which is a protein that promotes clotting, which we definitely need. Every time we get a cut, if we can't form a clot, we're just going to continue to lose blood. Well, some people don't make factor VIII, and now we are also able to safely produce this protein using bacteria, and it will eliminate the risks associated with donated blood, because instead of having to donate blood to people who don't have this clotting factor, people are able to just take the clotting factor, and then they're not subjected to the possible diseases that are transmitted by blood and the transfusion, or the possible problems with genetic information interacting with their genetic information by receiving blood transfusion. Here are some examples of genetically engineered drugs. So basically all anticoagulants that we use are genetically engineered. Those are all of the things that we add to overcome the problem where we need to dissolve blood clots. They're used to treat heart attack patients. Also, all colony stimulating factors. So these are the proteins that stimulate white blood cell production. They are genetically engineered, and they're used to treat infections and immune system deficiencies. Erythropoietin is responsible for stimulating red blood cell production, and it's used to treat anemia in individuals with kidney disorders. Also, that factor VIII that we talked about, which promotes blood clotting and is used to treat hemophilia. Not everybody has an adequate amount of growth factors, which stimulate the differentiation of growth in various cell types. So that's used to aid wound healing and support cell replication. The human growth hormone is genetically engineered and used to treat dwarfism. Insulin, like we just talked about, is involved in controlling blood sugar levels and used to treat diabetes. Also, interferons will disrupt the reproduction of viruses and can be used to treat some cancers. And interleukins, which activate and stimulate white blood cells and are used to treat wounds, HIV infections, and cancers and immune deficiencies. So all of these are genetically engineered drugs. Even though it sounds kind of futuristic to talk about genetically engineered things, it turns out we've been doing this for many, many years, and it's a part of our everyday life, and these genetically engineered drugs have saved a lot of lives. Another really interesting way we are able to genetically engineer things to overcome disease or viruses are subunit vaccines. So anytime you see piggyback vaccine or subunit vaccine, those are the same thing. This is like the nickname. This is the more technical term. Whenever we're talking about a subunit vaccine, those are like vaccines that are used against things like herpes and hepatitis. And basically, engineers will splice together the genes from the coat of the bad virus that's infecting someone into a fragment of a harmless virus, like cowpox virus for us, which will be used as a vector to carry that information into the person, so that now they have the ability, their immune system has the ability to immediately recognize that dangerous virus because it received that fragment from the spliced genes from the code of the virus. The recombinant virus will be injected into humans, and it's going to cause that production of antibodies, which is going to basically make it to where they can overcome this virus. This method of using one virus as a vector or carrier to introduce fragments of the DNA of a disease-causing virus is what we call piggyback vaccines or subunit vaccines, because the genetic information from one virus is piggybacking onto a harmless virus so that it can be carried into our cells, so that our immune systems can quickly recognize the dangerous virus if it's introduced in the future. So let's look at that with these images to help us understand. So here we have our herpes simplex virus. Here's the ring of DNA inside that virus. If we took this whole ring of DNA and put it into a cell, and then put that into a person, then we would have given them the herpes virus. But if we just take part of the DNA, enough to recognize the herpes simplex virus, and insert it into the harmless DNA of the cowpox virus, then suddenly we have introduced this recombinant DNA. So our harmless cowpox virus is carrying the genetic information to recognize the herpes virus. We put that into a cell and inject it into a patient. Now the harmless virus with the surface-like herpes simplex will be injected into the human body. Our antibodies will be directed against herpes simplex viral coats and then bind to the herpes simplex viruses that enter the body. The viruses will then be destroyed by the immune system. So basically, all we're doing here is taking enough of the genetic information so that we introduce the non-harmful parts of the herpes virus to this immune system, so that in the future, that immune system can recognize and immediately kill any herpes virus that's introduced. We do that using a vector, which is anything that is a carrier is something that you can call a vector. So this cowpox vaccine DNA is our vector to introduce that segment of the herpes virus. So the immune system will recognize it and kill it very quickly in the future. Because if we don't do that, the herpes virus will come in and our immune system will take a long time to figure out it's a bad guy. Usually the virus will have replicated so many times and caused so much damage that it's much harder to kill. But if we can recognize it right away at the first interaction of our cells with that herpes simplex virus, then we have increased our ability to fight that virus and to overcome it. So that's our subunit vaccines, also known as piggyback vaccines. We also have really interesting discussions of genetic engineering in this chapter. So we discussed engineering crops to be resistant to insect pests, which reduces the need to add so many insecticides to the environment. So you can imagine if we want to kill all the insecticides that are competing with us for our crops, we have to apply a lot of chemicals to the area to kill those insects. One way to try and overcome that is through genetic engineering of the crops. One example is using a soil bacterium gene from Bacillus thuringiensis, which we call BT for short, which produces a protein that is toxic when eaten by crop pests. These genes have been inserted into the chromosomes of tomato plants, and because the plants can synthesize BT protein once that gene is inserted, they'll become toxic to the pests that like to eat them, like the tomato hornworm. So that's one example of genetic engineering to kill insects with the genes you have inserted into your target crop. But we also have examples of herbicide resistance that has been introduced genetically. In herbicide resistance, we are genetically engineering our crops to be resistant to glyphosate, which is a powerful herbicide that kills most actively growing plants and is commonly used to control weed growth. So glyphosate is something that we will typically apply to weeds and try to keep off of our crops. But what we have figured out is that we could insert genes using a gene gun into our crop plant cells, and an isolated gene from a bacterium that we have chosen, which is resistant to glyphosate. So basically, take that gene that is resistant to glyphosate from the bacteria that are unaffected by that insecticide, insert that gene into our target crop in the cells, and now suddenly our crop plant will be unaffected by the application of glyphosate, which means we could potentially apply glyphosate to the whole field, and it's only going to kill or slow the growth of the weeds. It's not going to have an impact on the growth of the target crop plants. So this is what it looks like. It really is like an actual gene gun. It's shooting the genes in. We have DNA-coated particles, which will be placed into the gene gun and then shot into the plant cell. The main goal is just to make it past that plant cell wall, because once the DNA is in the cytoplasm, the cell will recognize the genetic information and the fact that it should be in the nucleus. And then that genetic information will be taken in through a nuclear pore and combined with the DNA that is already part of that plant cell. And then we have recombinant DNA. That's how we inserted the gene so that now these crops will not be affected by the application of glyphosate, and the weeds will die, but they will persevere. The real promise of genetically modified plants is to produce crops with desirable traits that directly benefit the consumer. So one example is so basically the ones we just talked about, we're trying to keep our crops alive. We're competing with flood, and we're competing with drought and fires. We're competing with animals. We're competing with insects. So we're just trying to provide a way where we can end up with enough crops for the people that need to rely on them. But another goal within genetically modifying food that we eat is increasing nutritional value. One example of that is the observed micronutrient deficiency in a lot of the country's major rice-eating countries. So in a lot of countries where the diet consists heavily of rice, we've observed deficiency of both iron and vitamin A. And scientists have been genetically engineering a kind of transgenic golden rice, which contains genes from a bean, a fungus, wild rice species, and a daffodil, with the goal of increasing its nutritional value. So basically, if we could provide a rice that supplies iron and supplies vitamin A, and the people will not have to adjust to growing a new crop or learning how to cook a new crop, they're just continuing to make rice, but it now has a higher nutritional value, then there's the benefit. Here's the genes they inserted. So they inserted the ferritin protein from beans, which increases the iron content itself. They also inserted the phytase gene and the metallothionein gene, which are involved with the uptake of that iron. So if you ingest iron, but it just passes through as waste and you don't absorb it into your system, it's not very beneficial. But these things will help the uptake of iron so that we are actually absorbing it and benefiting from it. Also, the beta-carotene gene, which is a precursor to vitamin A, we insert that gene so that vitamin A can be synthesized based on the ingestion of this rice. And that's a way to increase the nutritional value. So genetically modified crops are very commonly cultivated in the United States. We tend to grow a lot of genetically modified crops here. That's not true for every country. There are differing perspectives. Some of the benefits of GM crops include increased soil preservation and also reduced pesticide usage. So if you're inserting genes that makes the pest die when they eat your crop, then obviously you don't have to put as much pesticide chemicals on the ground. They're in the plants themselves, and they're supposedly, hopefully, only affecting the lifespan of those insects. But questions do come up about GM foods, and that makes sense. It's such It's such a new development in the course of human history and science itself, and it takes a lot of years for us to gather enough information to fully understand any new process in science. So some questions that come up include whether or not eating GM foods can be dangerous. So does adding genes introduce novel proteins that may be potentially harmful when consumed? And that makes sense. It's a fair question. If we introduce these genes with novel proteins, but we haven't ever had an experience with how to interact with them, because we, for example, with BT, we're not trying to eat the bacteria that are in the soil. We don't have a lot of interaction with that gene. But now we're interacting with the gene because we put it into our crops. So what if there are unintended effects that we don't realize, that haven't come to light yet? Another possibility is, what if we introduce proteins that become allergens? So it's not scientifically proven, but there has been talk about whether or not this rise in gluten intolerance is a result of all of our genetically engineered crops. What if the gene that we inserted that kills pests who eat it also affects whether or not we are allergic to the wheat that suddenly is giving us digestive problems? So that's another question. And then in the US, all introduced proteins have been extensively tested, and we haven't found enough scientific proof that GM crops are detrimental in any way to be ingested as humans, but the research will continue so long as we continue to use them. But the US has summarized that the risk of genetically modified foods is does not outweigh the benefit. The risk seems to be slight. There are other countries that disagree on that, but all of our research continues. One example that I wanted to mention about our genetically modified crops real quick is our cotton crops, which are commonly attacked by the cotton bollworm, budworm, and other lepidopteran insects. So in that specific class of insects, are heavy consumers of our cotton products, and more than 40% of all chemical pesticide tonnage worldwide is actually used just for cotton plants. As a result, we have introduced a form of the BT gene that is toxic to all lepidopterans, all of those insects, but harmless to other insects. And we have transformed cotton to a crop that requires very few chemical pesticides to be applied. Actually, another shocking number here is that 85% of all U.S. acreage for farming is comprised of BT cotton. So we are a huge producer of BT cotton in this country. Other questions that come up are the possibility of unintentional harm to other organisms. So weeds might be important sources of food or possibly shelter for those non-pest insects. The insects that we're not trying to kill and that we're not competing with, suddenly they are used to having a home in their ecosystem in those weed plants, and the weed plants are nowhere to be found because we've applied glyphosate, and though our crops are resistant to glyphosate, the habitat is suffering for the other insects that are not in competition with us for crops. Another question is the question of resistance. So as we know, as time goes on, bacteria, humans, viruses, everyone has the ability to develop this resistance to engineered proteins, to proteins in general. It's why we have to continue coming up with new kinds of vaccines for different strains of the flu, because resistance is something that is happening in genes of all species. And the question becomes, what if the pests become resistant to those engineered proteins? And now suddenly we've introduced these genes that are useless because, who knows, 10, 50, 40 years later, now the pests have developed resistance and they're unaffected by the gene, eating those crops just as much as they ever did. In a way to try and combat this, farmers are required to plant some non-GM crops alongside the GM crop in order to slow down the selection pressure for that resistance. Because if you've got one field of crops that's killing all of the insects, all of your insect buddies that are trying to eat it. And then you've got a field right next to it with non-genetically modified crops that do not harm you. The insects are likely to flock to that non-GM crop, and then you're slowing the pressure for that resistance to build in the offspring of the specific crop that you've planted. There's also a question of whether or not genes will be flowing into neighboring plants through cross-pollination. So what if our modified genes that we've intentionally put into our crop are suddenly showing up in adjacent plants that have interbred with our crops? How do we control that? How far does that spread go? These are questions yet to be answered. So that's on genetically engineered crops. We also have a very interesting discussion of reproductive cloning and the topic of therapeutic cloning in this chapter. So in 1938, Hans Spemann proposed that cloning might be possible by removing the nucleus from an egg cell and replacing it with a nucleus from another cell. Attempts at cloning were made many years later by several researchers. Some success was obtained, but the only success was with a donor nucleus that was from an early embryo, not an adult nucleus. Keith Campbell is a geneticist that proposed that in order for a successful nuclear transplant to take place, both the egg and the donated nucleus needed to be in the same exact stage of the cell cycle. They achieved this by starving the cells so that they paused at the G1 checkpoint, and then researchers actually ended up succeeding in cloning farm animals from advanced embryos. The first example of this was Dolly the lamb, who was born on July 5th, 1996. And basically, Campbell's colleague, Wilmot, attempted to transfer a nucleus from an adult cell into an enucleated egg. Whenever you see enucleated egg, that just means an egg with a nucleus removed. Wilmot used an adult sheep's mammary gland as the source of the nucleus. So they took the mammary gland cell, they removed the nucleus, and that became the nuclear donor. Both the donor mammary cells and the enucleated eggs were first starved, and then they were subjected to a brief electrical shock, which allowed the contents to fuse together. So basically made it to where the egg would accept the nucleus. The resulting embryo developed into a blastula and was then implanted into a surrogate mother. As a result, Dolly the cloned lamb was born on July 5th, 1996. And this is what that looks like. Here you can see we took a mammary cell from this sheep. This has the nucleus that is our target information that we want to keep. So we'll take that nucleus out. We'll take an egg cell from this sheep, remove the nucleus from it. It has the genetic information we're getting rid of. And then insert the nucleus from our target sheep. Suddenly, we will subject that to electrical shock so the contents fuse together. Those cells will start to replicate and divide and create an embryo. That embryo will then be inserted into a surrogate mother. And we will end up with a baby sheep that has the exact same genetic information as this sheep that we took the nucleus from the mammary cell from. It does turn out that most of these cloned animals do have different lifespans. They're usually shortened and they might have some genetic problems as well. Cloning procedures have become increasingly efficient despite the fact that they usually don't have a normal long lifespan. It's actually become so efficient that we have now developed a path for cloning your family pet. A lot of celebrities have been known to do this. I'm pretty sure the number in the book was about $60,000 to clone your dog. So that would basically mean that you will end up with a puppy that has the exact same genetic information as the dog that you knew and loved. But a problem with cloned embryos could be that as mammalian eggs and sperm mature, their DNA is going to be conditioned by the parent through a type of reprogramming called epigenetics. And then chemical changes are made to the DNA that alter when particular genes are expressed. So genes can be locked in the off position by methylation, where a methyl group has been added to some of the cytosine nucleotides. And these could cause problems. When we talk about stem cell therapy, we're usually talking about embryonic stem cells. So embryonic stem cells are the special cells that form really early on in development. And they're really special because they're totipotent, which is one of possibly the best words in biology, I think, because it matches so well with the meaning. Totipotent, like it has total potential to turn into anything. And that means that the cell, an embryonic stem cell, has the ability to form any kind of body tissue, any kind of cell, even an adult animal. So that's why these embryonic stem cells are so sought after, and we have a lot of research around them. We also all have adult stem cells, which are pluripotent, which means they can turn into many types of different cells, but not all of them. So you're definitely going to want to remember that. Embryonic stem cells are totipotent. They can turn into any kind of cell. Adult stem cells are pluripotent. They can turn into many different kinds of cells. Pluri like plural, many different types of cells, not total. As development proceeds, some of those embryonic stem cells are going to become committed to forming one certain type of tissue. And every major tissue is formed from its own tissue-specific adult stem cell. So basically, we all have embryonic stem cells. Once we get old enough that they're specialized to produce one single type of cell, maybe cardiac cells, or digestive cells, whatever cells they're specialized to create, then they become adult stem cells. Because embryonic stem cells can develop into any tissue, they offer an exciting possibility of potentially restoring damaged tissues from many different kinds of conditions and many different organs. Here's how it looks. So we'll take a sperm and fertilize an egg. As a result, we're going to end up with this embryonic stem cell mass in our blastocyst. If we leave that blastocyst alone, it goes on to become an embryo. If we instead remove the embryonic stem cells and grow them in culture, then they could potentially be used to be inserted into a patient and help them overcome whatever disease they have. Maybe they have heart problems and they need cardiac cells. Maybe they have problems with their vision and they need new eye cells. Whatever the cells may be, those embryonic stem cells would be able to be inserted and then differentiate into those types of cells that are the problem area. However, there is an ethical dilemma that goes along with this, because some people believe that once the sperm fertilizes the egg, this is a living organism. And they believe that it's not right to take the cells from this living organism to heal this person. However, the other side of thinking is that this is a mass of cells that could potentially save a life. It's not a living organism until it can sustain life on its own, and therefore you're not really harming anyone by harvesting these cells to cure patients with multiple different problems. That's going to continue to be an ethical dilemma so long as this is present in science. Experiments using stem cells to repair damaged tissues have been carried out in mice without functioning immune systems. And for stem cell therapy to work in humans, the main problem that we have to overcome is immune rejection. And immune rejection just basically means that we could try to insert these embryonic stem cells to solve a problem. But then that patient's body is going to recognize it as a foreign something, a foreign threat. It doesn't have the nucleus, it doesn't have the genetic information of the patient. It seems foreign, it seems like a threat. The immune system is going to work to kill those embryonic stem cells before they can really help the patient. One way to try and overcome that is with therapeutic cloning. which is also known as somatic cell nuclear transfer. And this is a way of trying to address that issue of immune acceptance. In therapeutic cloning, the nucleus from that patient is going to be inserted into an enucleated egg cell, where the nucleus has been removed, and then we'll let that egg develop in vitro. Once we have a blastocyst, the embryonic stem cells can be removed and then used to replace the patient's damaged or lost tissue. So basically, because we took the nucleus from the patient, now when we insert the embryonic stem cells, they're recognized as their own cells. They have the same genetic information. So now the immune system is not triggered to immediately kill those embryonic stem cells. And then they could actually differentiate into the cells we need and help the person. As I said, many people regard therapeutic cloning to be ethically unacceptable. There will always be this divide. Reprogramming adult cells to become embryonic stem cells is something that we're looking at as a potential alternative approach. So basically, can we take adult cells and modify them so that they will become totipotent, the way embryonic stem cells are, without having to harvest embryonic stem cells from a blastocyst? So that process looks like this. Normally, an embryonic stem cell introduction, without the use of therapeutic cloning, you would just grow those embryonic stem cells, put them into the person. The immune system is very likely to then attack the embryonic stem cells before they can be of any benefit. But if we take the nucleus out of a regular skin cell from a patient, and then insert that into an enucleated egg cell, and then allow it to become a blastocyst, then at that point we can harvest those embryonic stem cells, grow them in culture, and insert them into that patient so that the stem cells could develop into whatever cells that patient's needing. In this case, it would be pancreatic cells to help them with their diabetes. And that is a way to overcome immune rejection and work towards immune acceptance through therapeutic cloning. At this point in the process, if we had not harvested the embryonic stem cells and grown them in culture, and instead just let that blastocyst continue to progress, then this would have been reproductive cloning, because we introduced a new nucleus, it became a living organism. But because we're doing this cloning, this therapeutic cloning for the purpose of healing a patient, that's what we have with therapeutic cloning, where we are harvesting the embryonic stem cells with the nucleus from the patient to overcome immune acceptance, or to overcome immune rejection and lead to immune acceptance of those new embryonic stem cells. We also discuss gene transfer therapy in this chapter, which involves introducing healthy genes into cells that lack them, or even cutting out genes that have been mutated or genes that are problem-causing and replacing them with genes that are beneficial. There was early work with a cold virus vector called the adenovirus, which was unsuccessful in humans because of that same problem with immune rejection. And as a result, we've started using a smaller vector that is less problematic, the adeno-associated virus, because it does not elicit a strong immune response, and so far the results seem promising. Here's an example of gene therapy, where we are introducing new genes to cure some kind of disorder or disease. In this case, we've got our adeno-associated virus acting as the vector or the carrier for the gene we're trying to insert. Here's our healthy version of the retinal gene. And this is the gene that dogs that have good vision will tend to have. If they don't have this gene, or if they have a mutated version of the gene, they could be blind and unable to see completely because they're not producing that specific protein that is needed for them to have good vision. So we will take the viral DNA from that adenovirus and insert that specific target gene that the dog does not have. After combining the healthy gene with the viral vector, we will have this recombinant DNA inside our virus, which can then be injected into the viral space, into the retinal space within the eyes of young dogs that are known to have this degenerative disease. As a result, the young dog with that retinal degenerative disease will begin to make that protein, which is a product of that gene that was inserted, and you will observe restoration of vision. So now suddenly the dog is able to see because it has the gene to produce that protein that is needed for vision. CRISPR is also a really exciting new technology that has been able to allow scientists to edit genes in a really specific way. This technology is based on an unusual repeating DNA sequence that is found naturally in bacteria. Basically, there are several dozen base pairs that will be followed by the same sequence in reverse, and then 30 bases of kind of spacer DNA that are just there to provide space between these segments. It turns out when that section of DNA that is repeated in reverse is transcribed, it's going to produce a piece of RNA that's going to fold back on itself because of the affinity between those nucleotides. And then that 30 base spacer will end up forming a loop. The RNA loop is going to bind to DNA cutting enzymes called endonucleases and to sequences of the genomes of different viruses that will attack that bacteria. In nature, bacteria uses the CRISPR sequence to attack viruses by recognizing and cutting up their DNA.